Welcome to Global Expert Talk, an initiative of Voice of Healthcare on variant calling, annotation, and interpretation, making sense of NGS data. Next generation sequencing generates vast amount of data, but its true value lies in accurate variant calling, robust annotation, and thoughtful clinical interpretation. Translating raw sequences into meaningful, actionable insights requires bridging informatics, molecular pathology, and clinical expertise. To ensure NGS results truly inform diagnosis, prognosis, and patient-centered care. Today we have with us Sarath Babu Krishnamurthy, who brings over a decade of experience in genomics, bioinformatics, and healthcare innovation. Currently, he is the Director of Genomics, Health and Informatics for the United States. Welcome, Sarath.
Thank you.
Right. So I just need to understand that what actually happens after cell NGS testing.
Okay. NGS is next generation sequencing. Just giving you a background of where it all started is in 2003. That was the first time that a human genome was sequenced. A whole genome was sequenced, and at that time it took billions of cost. And each nucleotide by nucleotide it was sequenced. And then over the years, the technology has grown, and then more high throughput sequencing technologies grew. And now we have the capacity to sequence a genome in a day. Previously, which took years to make, now we are able to sequence a whole genome in a day. And this advancement happened because of the next generation NGS technology, where high throughput sequencing is possible. And so the sequencing can be done at multiple levels. So it can be either looking for a single variant in a single gene at a particular location, or it can be a set of genes that we are looking for, or it can even be like an exome where we are looking for only the coding regions of the genome. And then there is the whole genome where we sequence the entire 3 billion base pairs of a particular individual. So all these things are possible. So depending on that, the backend NGS workflow differs. But basically, it all starts with someone getting your either a blood sample or a saliva or a tumor tissue sample. That's all, or frozen tissues. It could be all these different varieties, and then it goes through different steps. There are 4 different steps that happens. The first one is the DNA extraction, and whatever the sample that comes, the DNA needs to be extracted. And then once the DNA is extracted, next it goes through a library preparation, where that particular, depending on what is the goal, whether it is a single gene confirmation, or is it a whole exome or whole genome, depending on that, the library is prepared. By library is prepared, I meant like the given DNA is being fragmented into 150 base pairs, 150 to 300, or if it's long read sequencing, it could be up to 1000 base pairs. Then all these fragmented DNA are attached with adapter do all these, then go through a sequencing machine. This is the third step. DNA extraction. Second one is the then the actual sequencing. So let's take, for example, Illumina sequencing. It is a flow cell by cell synthesis platform. So all these extracted DNA then are put into the sequencing machine, where it actually amplifies the given DNA fragments. And then for each base pair, it can amplify it up to 300 to 500x in that particular location. And if it's genome, we don't prefer that high because it will get much costlier. It can be up to 30x, 30 times amplified, or 500 times amplified if it's, especially if it's a tumor specific place that we actually. Are just small chunks of DNA fragments that are sequenced and multiple times in the same location. and all these need to be put together in context to understand it. So that's where the bioinformatics analysis comes, where we aggregate to one reference genome. We know how the human genome looks like because of the sequencing effects that drivers that happen. And then over the years, more people have been sequenced, and we understand that, oh, these are population specific variants. So we get a linear structure of the genome, and then we try to align a new sample sequenced to the same linear reference, and then try to understand where are those positions where there are changes that are happening. So,
Right.
yeah.
Right. So we have the library and we have the reference genome with it is compared, and maybe that helps in annotating and finding out the actionable, mutations. So what type of genetic changes does NGS look for, and which one are the most relevant for patient care?
So NGS looks for anything that is really small to a large changes that are happening in the DNA. The small, it could be a single nucleotide change. These are called the SNVs. So the SNVs happens in just one location, and then there are small indels where one to up to 50 base pairs. It is called small, where the changes could happen, and then there are large events like CNVs, copy number variants, where you either delete an entire region of a gene or even a full gene, and then or it could be duplicated or it could be inverted. All these type of events are possible. These are the different types of variants that are there. And then what is critical for the patients is understanding the context of where these variants occur, whether it is impacting the function of the protein, and could that lead to change in the phenotype of the patient, where because the protein is not functioning, then what happens in the patient. So that is the critical context that need to be understood in order to identifying or making sense of all these that we
Right. Changes that are happening, that is also very important because that results in whether a particular protein is being affected or not. So how does a laboratory decide that which genetic change are important and which can, ignore?
So we get all these depending on which panel that we do, whether we look for a specific set of genes. And example, we would have decided that like this patient has a certain type of cancer, and so we took a particular tissue sample from that particular cancer type, and then breast cancer, and then let's say we are a breast tissue sample that is being sent for sequencing. And there are then when the sequencing is done, there are already a known set of genes that are affecting the breast cancer. So these cancer panel genes are then looked for any pathogenic variant in them. It could be SNV or Intel or CNVs. So depending on the context of which gene that we are looking for, and then those those variants in those genes are looked for multiple criterias. So it could be either a tier 1 variant where we know the pathogenicity of the variant, meaning it can we know the function of the gene, and then a change in this gene because of this variant could affect the function of the gene. Then that's the pathogenicity. And then next one is the penetrance. So not everyone who has a variant in a gene could actually end up having the phenotype. So it could just be a carrier mutation that it does not actually translate into a phenotype. So we need to understand, do 100% of the patients having this variant have the disease phenotype, or is it only 50% of them? So understanding the penetrance is an important one. And then the expressivity. So the expressivity explains why certain variants, actually the phenotypes of the patients, even if they have the same variant and even if it is a good penetrance, once the phenotype might vary, because the symptoms of showing up of a different phenotypes differs based on the expressivity of the variant. So all these things are taken into consider, and then if all three are looked upon and considered as an actionable one, then it is marked as a tier 1 variant, where there is a clinical trial that is actually possible for that variant. Because of the change, we could actually like give some medications that could actually reduce the work of this variant. And so we could actually make some meaningful clinical diagnosis for the patient. So that's tier 1. And then there is tier 2, which is, we know it is pathogenic, and we know some understanding of the penetrance, but we don't know the exact expressivity pattern, or sometimes even the penetrance is not known. Everybody, like, there are new mutations that are being found all the time, and all these variants we need to understand in the general context of healthy people, whether healthy people have these variants. So that's where the population databases come into play. They help us understand whether do all these people who have this variant have a disease, or is it common in the population. So that helps us understand that. And then the family history plays a major role in understanding as well. Have they been family members who also have this variant and was affected with the same phenotype? So understanding that actually helps. But then without all these information, we won't be able to classify the variant as tier 1 and drug target being available for that. So in that case, these are all classified as tier 2. And then there is tier 3, where we don't know enough biology about the variant, like whether that variant itself actually makes an impact on the gene or not. Some genes are studied over the years, and we have enough information about the variants in there and how they function, which pathways they belong to. All those information are possible. But there are new genes that are being identified, and it takes a long time, like 10 to 15 years, to understand about all these different genes and the functions of every single variant that are in there. So these are called variant of uncertain significance. This could change over time, but right now we won't be able to make any meaningful action based on these variants. So these are classified as VUS and tier 3 variants, and then tier 4 variants, which are benign variants. Uh, Really, all of us have a lot of variants, and not all of them are actually pathogenic and that could cause disease. Many of them are just benign. Uh, the differences in us that brings those, that does not cause disease. So those are all benign variants.
So we were talking about the tier system. That is what you were telling, and you explained the variant of uncertain significance as well. Um, so very well explained. I believe we should know the tier system, and that helps us in designing the clinical trial as well, and knowing the penetration and expressivity, as you said, is also very important. So on that note, I just want to know that is there any age-related changes you find while preparing the library or comparing it with the reference genome, or are there any technical artifacts which you feel that won't translate into any actionable insights?
Yeah. So there are clonal hematopoiesis stem cell changes that could occur, and this is very common in elderly population, as people above 50 years of age generally have these changes happening. And these are like one of well-known there are well-known chip genes which actually get those changes, and these changes could lead to more chances of having cancer, or even uh, cardiac arrest and other phenotypes, but they are not the driver mutations for the cancer itself that we are whatever phenotype that we are looking for. So understanding the difference between a tumor mutation, which is a somatic mutation found in a tumor cell, to a mutation that is identified in a chip cell, right, in these genes. So understanding the differences in them is actually very important, and it's very rare to identify as well, because there are certain genes like TP53, which is a well-known cancer gene and also is a chip gene. So the understanding where the mutation occurs and then understanding the variant allele frequency, we might end up getting some blood cells also in there. This could genes, and so we might identify them as a somatic, just be a chip variant. So understanding which gene it occurs and then what is the actual phenotype that we are looking for, and is it a common or rare one, and understanding that is very important in identifying that. And the artifacts could occur at multiple levels in order to understand. So it could be as simple as like how much DNA that was sent in, and is it good enough quality? Are there any contamination in there when the sample is being sequenced? And like in the bioinformatics analysis also, there are a lot more checks that could be done, whether were there a sample swap that happened, or it could be the coverage of the gene. So we would expect the coverage to be really high